arose de novo suddenly, and then in all species except for a few, it kind of got scrambled again because it wasn't used.
So that would actually be not an example of the novel origin. That is, this in a situation like this, all the species would have the gene, but it evolves so fast that you can recognize it only in some. What you need to prove is that the gene really originated somewhere, in a species very recently and has originated nowhere else.
Exactly. Okay? So the challenge is to scenario I described to rule it out.
Exactly.
Right.
And for that, you usually need experimental data.
Yeah.
We're good again?
Yeah.
Okay, thank you. So you need experimental data to confirm that such genes are really de novo genes. But the question then would really be, even if you have proven that, how could it be that something that's transcribed and translated, indicated here by the yellow and the blue structure, the blue symbols here, has actually originated de novo, and how could that complexity arise in a single step? Well, the answer again is like what Darwin already didn't know, but we know now, is that you can break down these this complex object into simpler ones, and their origin can be independently useful. And I'll just illustrate what I mean with this slide. Okay? It turns out that something that Jacob didn't know at the time, but we know now, is that many genes actually don't encode proteins. Non-protein coding genes are only transcribed into RNA. So this is not just messenger RNA, but it's a different kind of RNA. In fact, different classes of RNAs called microRNAs, small nuclear RNAs, and so forth, that don't really matter for our purpose here. And these genes, these RNAs have specific functions in a body. For example, they may help regulate the expression of genes, of other genes, for example, turning them on or off, or they can modify other molecules chemically. And what you see here on this slide is an overview over non-protein coding genes in the human genome. If you add up these numbers, that add up to probably more than 7,000 or 8,000. And considering that the human genome has only about 20,000 protein coding genes, that's actually a substantial fraction of all genes in the human genome. So this illustrates that you could actually have an origin of genes de novo, something like this, where first you get a gene to be transcribed, DNA to be transcribed, and then perhaps only later translation is added onto this. Okay? So in principle, there is two ways to study the de novo origin of genes and individual parts that are necessary for them to function properly. And the one is something I already mentioned, namely by computationally and comparatively, that is, by comparing many genomes, and especially closely related genomes, to detect cases like this. As I said, this needs to be supplemented by experiments. Another approach is based on experimentation. So experimentation here is the foundation of this approach. And what you do for that in this approach is you introduce mutations into DNA that does not already encode a gene, and ask how often do such mutations create a gene or one of its elements. And this approach with the technology that I'll be describing can be actually done in fairly high throughput, with many mutations. And when you do that, you can actually collect statistics such that the statistics can help you elucidate principles of gene origins. For example, you may be able to ask whether some kinds of DNA are especially likely to give rise to new genes. So this is the approach that I'll be talking about today. And in particular, I'll be focusing on what is perhaps the most important and most elementary part of a gene, namely the transcription, the signal to start transcription. And in bacteria, the signal is called a promoter. Now, if this horizontal line symbolizes part of a genome or a stretch of DNA, and if this arrow indicates where transcription starts, so these wavy lines correspond to messenger RNA molecules, and +1 would be the coordinate by convention, it designates the first letter of DNA that is being transcribed. And close to that start of transcription, there's basically two elements, DNA words, if you will, that need to be in place. The first is called a -10 box. And this minus 10 box is so called because it's 10 nucleotides upstream of the start of transcription. And it basically has allows a particular spectrum of DNA sequences that make it a minus 10 box. Likewise, we have a minus 35 box that also emits a particular spectrum of 6 mers on DNA that make it a minus 35 box, and these need to be spaced approximately at 70 base pairs apart. Okay? So this is the essence of what a bacterial promoter, bacterial transcription start signal is about. Now, if you show this to a computer scientist, they will immediately say, oh, this is a trivial problem. Right? You know, if I give you a DNA sequence, if you give me a DNA sequence, I can compute for you the probability that it contains this combination of boxes at this distance, and perhaps one could even compute that analytically. But it turns out, and this is where sort of theory hits the limit, that this has been tried and has been tried for about half a century. And with rather not much success, really. So it turns out computational methods, you know, even after more than half a century of research into DNA, are not good enough to predict something as simple as this and whether it's actually functional when you put it inside an organism, that is, whether it really triggers the start of transcription. There's a number of reasons for that. One of them is that, for example, the sequence context, that is the surrounding nucleotides, ways that we don't fully understand, and these requirements for specific words don't seem to be strict. For example, we know some bacterial promoters that don't have a minus 10 or a minus 35 box, or neither, and they still function properly. So what we need here is not just theory and computation, but we actually need experiments to see how likely it is that something like this occurs in DNA or arises in DNA. And I'll tell you about some such experiments next. Now, another piece of background information, though, before we go there, is that a promoter can be weak or strong. That is, a weak promoter will drive very little transcription, that is the production of little messenger RNA per unit time, and a strong promoter will drive a lot of transcription. And finally, what I've told you about now is only the simplest kind of promoter called a constitutive promoter, which can drive transcription on its own. But there's other promoters too. For example, some promoters require activation. And this activation involves a molecule, a protein molecule that's called a transcription factor or a transcriptional regulator or a transcriptional activator. And such a transcription factor binds a specific DNA word on the near the promoter. And when it binds there, it helps the biochemical machinery that is responsible for transcription, that's called RNA polymerase. It helps the RNA polymerase recognize these boxes, bind there, and then start transcribing. So that's an important principle of promoter regulation. Other promoters turns out, are subject to the opposite kind of regulation that's called repression. That works via a transcription factor that is called a transcriptional repressor. When that one binds, it prevents RNA polymerase from recognizing this and starting transcription. Okay. So that was all background you needed to know before we can talk actually about the experiment that we performed. And this experiment turns out to have two parts that I'll talk about in order. The first part concerns the identification of active promoters, that is, promoters from which transcription occurs. And only the second part deals with creation of promoters de novo. It may not make sense why we need this part, but it will make sense hopefully a few slides down the line. Okay. So what I'll describe next in fairly simple general terms is how this experiment works. Okay. So it first starts with the creation of many random and synthetic DNA sequences. So these are DNA sequences that we produce in a DNA synthesizer that have never seen the inside of the organism. There are 150 base pairs each long. The reason is that this is the typical length of a piece of DNA that regulates gene expression in E. coli. Our experiments will be performed in E. coli, and it has a approximately equal contribution or frequency of each base or each letter or each nucleotide at each position of about 25%. There's a distribution around that, but the average is 25%. So we start with a collection of many such sequences. In particular, in this experiment, we used about 17,000 of them. And then we placed each sequence into something that's called a reporter plasmid. So a plasmid is a circular piece of DNA that we can stick inside a cell that is able to replicate on its own in a cell like that of E. coli. And this plasmid has two parts that are really important for how it works. And these parts are called reporter genes. They're shown again here, a bit magnified in a linear dimension of a circle, like on the plasmid. The first of these genes, called GFP here, encodes a protein that's called green fluorescent protein. And when that gene is expressed, that is when its DNA is transcribed and translated, cells will emit green light if we expose them to laser light of the right wavelength. And then the other gene is called RFP. It encodes red fluorescent proteins that when it is expressed, cells fluoresce in red. Now, what these genes don't have is that they both lack a promoter. Okay? So actually, what I didn't say, I should have explained this. So these genes are in opposite direction on DNA. Remember, DNA is a double-stranded molecule, so we could, in principle, have a gene encoded on each strand. And here, this gene would be encoded on the top strand if we drew it horizontally as a double-stranded molecule. And this would be oriented on the bottom strand being transcribed in the opposite direction, because a promoter could actually drive transcription in either direction, depending on which strand it occurs. But when we start out with this plasmid, these genes don't have a promoter. So the DNA between them, shown as this black box, is inert. It does not drive transcription. And then what we do then, as we say in the jargon of molecular genetics, we clone this entire collection of random DNA into this reporter plasmid in between these two genes, such that we get a huge collection of plasmids where different plasmids harbor different inserts or different random sequences. And we then basically ask the question, well, do some of these random sequences perhaps drive transcription? But at this point, we're not ready to do so yet, because we only have the plasmid itself and the DNA inserted into it. The plasmid needs to be inside a cell such that gene expression can occur. And so this is the next step here. We basically stick this whole collection of plasmids, which is also called a library of plasmids, like a library of picture books, different kind of library, a library of DNA sequences. We stick this library into E. coli cells such that different cells would then harbor different library members. And if any one such insert or member of the library can drive gene expression, that is, when it's a functional promoter, it will cause cells to fluoresce either green or red, depending on the direction of transcription. Yes?
How does this work in practice? Do you have like 17,000 Erlenmeyers?
No, it's one. That's the trick. You can do this all in one reaction vessel. Yeah? So you basically take the whole collection of plasmids, the whole plasmid library. You have, a flask with billions and billions of them, okay? Even though there's only 17,000 different ones. And you expose them to E. coli cells that are prepared in the right way so that it can incorporate foreign DNA.
But so actually, that it shows green, you only know that some work, you don't know which one.
Exactly. So that's, at this point, that will be on the next slide, how you find out. But just to give you an idea how this would look like, let's say I would take a sample of cells and plate them on a petri dish. And the individual cells would form colonies, so they would divide and form these blobs here. Each of these blobs would correspond to a large collection of cells that all descended from a single cell that harbors one library member. And you see that some of these dots are green, some are faint green, some are very bright green. And that tells us that, yes, there's probably something happening in there in the sense that the green fluorescent reporter gene is being expressed. However, it turns out plating these cells on a petri dish is not really quantitative enough or useful enough, especially when you want to do this experiment on a very large scale, partly because you can only resolve about 1,000 cells or colonies on a petri dish, whereas we need to resolve many more. So we use a different kind of technology that involves DNA sequencing that works as follows. The technology is called sort-seq. It's sort of widely used in this area of high throughput analysis of DNA. And it works as follows. It starts with a fluorescence activated cell sorter, or also called a fax machine, not to be mistaken with the fax machines of old times that people don't use anymore, that transmit information. But it's a machine that basically allows you to run a large population of cells through them, and it will examine each cell. And depending on how the cell fluoresces, it will set the cell aside or not. And more specifically, so basically it will sort the cells. I'm sorry, this should say it sorts sequences, but it will sort cells into multiple bins according to the intensity of red or green fluorescence. And specifically, we used here 4 bins. 4 bins for GFP, that is cells that have no expression whatsoever of GFP, that is, they don't fluoresce in green, weak, moderate, or strong expression, and the same for RFP. So a total of 8 bins. So what you have after this step is you have basically 8 tubes that contain different subpopulations of cells depending on their red or green fluorescence. And what you do then is then you read the DNA sequence of only the inserts that you into the plasmid, only this part here that comes from here. And you read a lot of them, about more than 1 million DNA fragments per bin. And simply you count then the number of sequences of each of these fragments or each of these individual library members per bin. Now, what complicates this analysis is a little bit, and something I didn't tell you about so far, is that transcription is a very noisy process. That is, a gene that has a specific promoter, its transcription level, that is the amount of messenger RNA produced from it, will always fluctuate up and down a little bit. So, for example, if we would normally sort a gene into this, or a cell expressing a particular reported gene from a particular fragment into the weak bin, sometimes it might find itself in the bin of no fluorescence, sometimes it would find itself into the bin of moderate fluorescence. So we need to take account of this fact, and what you basically do is you calculate a weighted average of fluorescent intensity that is weighted by how often we find a particular sequence in each bin. And the result of that is basically data that looks like this. Here would be our list of 17,000 sequences. And for each of these sequences, we would get a quantitative fluorescence score that we scale from 1, meaning no expression, to 4, strongest expression, for either GFP or RFP. So in this hypothetical example, there is quite a bit of expression of GFP, so this would correspond to the moderate category, but very little expression of RFP. So this would contain somewhere in these 150 base pairs, not all of which are shown here, a promoter that drives expression of GFP, but not of RFP. Here you have the opposite situation. A lot of RFP expression, no GFP expression, and here you have a situation where both are expressed, although not at equal levels. So that's the idea behind this experiment. Looks rather easy in principle, but takes a year or 2 to get all the steps right. Yes.
Is it true that if you would not insert any sequence, that you would get a score of 0, 0?
Would get something that's close to 0.
Okay.
Yes. But there's as in any measurement instrument, there's instrument noise, right? You need to take into consideration. Okay. So here's already an important and very simple piece of data here. What's shown here is essentially a 2-dimensional histogram that shows on the horizontal axis green fluorescence on a scale of 1 to 4, 1 being nothing, 4 being maximal, and on the vertical axis red fluorescence. And these dashed lines indicate what we consider fluorescence that is significant, that is substantial enough that we call this DNA sequence to be expressed. And we chose these boundaries for consistency with other papers who studied this kind of process, just with different techniques and on a much smaller scale. And what you see here is that, well, the vast majority of these 17,000 sequences drive no expression, but then there is 27% that drive sequence the expression on one strand, and the other 4% on another strand. And there's only a relatively small minority that drive expression in both directions. And this, the number here that we reach when we sort of average over these categories is that about 16% of random DNA contains active promoters. And that is a really remarkable number. It's consistent, by the way, with recent smaller scale studies. What it tells you is that a random piece of DNA has a 16% chance of actually driving expression of whatever is next to it. So from this point of view already, Jacob, was really, and a lot of other people were barking up the wrong tree when they thought that it's really hard to produce the elements of a gene or entire genes because it's actually quite easy to produce random promoters that even occur in random DNA at a fairly high frequency. Now, when an evolutionary biologist, here's what I just said, they will usually, yes.
So is another thing you can conclude from this figure on the bottom right of the previous slide, that promoters are not such discrete things.
Exactly. Yes, absolutely.
Okay.
Yeah. And in fact, we know that, for example, a lot of genes that have been detected to originate in Novo in actual genomes start out being expressed at very, very low levels. Okay, they're almost indistinguishable from noise. And in this general area of single cell, large scale expression analysis, distinguishing noise from actual signal is one of the biggest challenges.
Is there a reason why there is such a high difference between?
Very good question. I get that one every time, and we have analyzed about this for a long time. And part of the reason is likely has to do with intrinsic differences in RFP and GFP. So, for example, one of them has a longer half-life, and that creates different signal-to-noise ratios. So it's a bit difficult to compare them, but we don't actually have a way of creating reporter genes that have exactly the same property except fluorescence colors.
So it's not. You don't think that the difference is caused by the fact that there are more start codons in
it shouldn't be. Okay. It shouldn't be because it's random DNA. It could be a sequence context phenomenon because RFP and GFP do not have the identical sequences. The problem is it's difficult to test because it's not something, so if you see this for the first time, you think easy to test, you basically invert the orientation, right? Yes. But you can't do that because this is based on thousands of sequences, okay? And you can't, invert them in an orderly way, at least with conventional cloning techniques. So this is an experiment that, at some point in the future, we'll have to think about how to test this, whether it's has to do with sequence context or with RFP and GFP properties, but it's not an easy thing to do. Okay, now an evolutionary biologist would say to what I've said so far, well, this is all nice and well, but sequence evolution does not take place in random DNA. It takes place in a genome, and the genome has properties, DNA sequence composition that has been shaped by millions of years of evolution, and that has very different properties of random DNA than random DNA. So you should actually do this experiment with genomic DNA. And so we did. What we did is we basically repeated exactly what I just told you about. But we took, instead of pieces of DNA out of a DNA synthesizer, random fragments of the E. coli genome of about the same length. And we took so many fragments, about 90,000 fragments, that each of the E. coli nucleotides would be covered about 2 to 3 times. And so we covered essentially the entire genome, except for a small region that is very difficult to clone. We created a plasmid library from these fragments, transformed E. coli cells with the library, and quantified expression in exactly the same way with sort-seq. And then asked, well, is it the genomic DNA or the random DNA that is more likely to harbor active promoters? And what we found is actually the genomic DNA. And what you see here is the proportion of promoters in the genomic library, the random library, that is stratified by what's called the AT content. That is the composition, the content of A or T nucleotides in DNA. And this is important because we know that promoters are rich in AT, and ignoring AT content therefore would have been an important confounder in our analysis. But it turns out, independent of AT content, genomic DNA is more likely to harbor an active promoter. Now you could say, well, this is kind of not so unexpected because the E. coli genome contains 4,500 genes, and all of these genes have a promoter. So these are probably just the promoters of the E. coli genome. But that turns out not to be the case. Because we have a lot of the advantages of doing such a relatively high throughput experiment, because we have a lot of different DNA fragments, we can restrict the analysis of the data to DNA fragments that come from particular parts of the genome. And in one analysis, there's many others that we've done in this paper that I showed, that's on the archive already, we restricted the analysis to only fragments that came from inside genes. So that are not the genes of the promoter of any genes. And it turns out the picture is essentially unchanged. That is, genomic DNA has sequence signatures that we do not fully understand, at least based on this data, that make them more likely to be active promoters. And I will come towards, well, in the next slide, but also even more so towards the end of the talk, to a possible explanation for this. Now, we don't understand all aspects of this data, but we have a candidate explanation at least. Now, an inkling of this candidate explanation comes from the following analysis. So, and this is an example of where you can actually integrate computational predictions with the kind of experimental data that this experiment creates. So, computational predictions are not without any value in this world. You can actually use them, but they need to be integrated with experimental data. So what we did here, we took advantage of the fact that the E. coli genome encodes many different transcription factors, about 300 or so. And for about 90 or so, we have sufficient information about their sequence specificities, that is, which kind of DNA sequences they like to bind, and thereby regulate nearby genes, to actually predict whether in our library of 150 base pair long sequences, to predict whether a factor might bind or not bind to such a sequence. And specifically for those of you who might care, we use a tool here that's called a position weight matrix that basically tallies up sequence preferences of such a binding site, nucleotide by nucleotide, for any one transcription factor. Okay? And what you see here in this slide is based on computational predictions. So it is the predicted proportion of sequences in our library with more than one transcription factor binding site. And this is stratified here by what's called the information content of a binding site. So what is the information content of a binding site? To specify a single nucleotide, a single letter in a 4-letter alphabet, like the DNA alphabet, you need 2 bits of information. So imagine you have a transcription factor that binds a 10 nucleotide strand in only A's. Just a hypothetical example. Well, you need 20 bits to specify such a sequence of 10 A's. If it were able to bind A and T interchangeably at each of these positions, well, for each A or T, you need only 1 bit. So then the total information content would be 10 bits. In other words, the information content of a binding site indicates how much information you need in bits to specify the binding site, and it's low for short binding sites or binding sites that are very degenerate. That is where many different nucleotide words can serve as binding sites. It's high for binding sites that are long and very specific. Okay? So what you see here from this data, by the way, these bands here indicate 95% confidence intervals. that just didn't translate well from Adobe Illustrator into PowerPoint. And they show that especially for low information content binding sites, random DNA contains more binding sites for transcription factors. And we think, well, we have no proof at this point, that this could be a partial explanation for why genomic DNA harbors more active promoter, because we just showed that random DNA is less likely to contain active promoters, but contains more binding sites. So this suggests that random DNA contains more binding sites that repress transcription. Okay? So I'd like to store that information away for a moment. Towards the end, I'll be coming back to it. Yes?
Can I ask you about that experiment that you did with the non-genomic DNA?
Yes.
So do I understand correctly that you take 150 base pairs from in between the promoter and the terminator? That's basically not a promoter.
No, we shred the entire genome in random places. Could be anywhere in the genome.
But you said it was like it was not including promoters.
Oh, and so you said about the random DNA.
No, so the
genomic
one that you
DNA.
showed actually was like where you take pieces of DNA that do not contain promoters.
Yes.
You put that in the plasmid and then it turns into a promoter. So how is
Sorry,
it
I have not, I don't think I have said, because it would not be correct to have said that this is DNA that does not encode promoters. We don't know what that DNA encodes when we start out. Eh?
So even if they are from the interior of a gene,
Yes,
it might be
exactly. That's correct. Yes.
So the previous slide showed that this random library is enriched with promoters. Is it not true that whatever other thing you choose, they will be depleted in that as long as that thing is disjoint from promoters?
So the question is, should we call the random DNA enriched or the genomic DNA impoverished in promoters, right? Right. And it turns out that for the correct interpretation is that the genomic DNA is impoverished in promoters. And I'll get back to that. Okay. Now, the question arises though, so I've been going now for about 40 minutes or so. We had a little bit of discussion. How long should I be going? Just to know when I should cut it off. Do we have an opinion?
1 hour, then we meeting for question.
Okay, well, then we started the questions already, so maybe I'll talk a little more and we'll have fewer questions at the end, okay? Okay. So this is now where I come to the second part of our experiment. And so the first part would still be a bit unsatisfying to an evolutionary biologist, except especially one that studies molecular evolution of DNA. Because they would say, well, this is really not what you've done, does not really reflect how new promoters might arise in a genome. The way new promoters arise in a genome is not that there's random fragments of DNA inserted somewhere in the genome, at least not usually, but that DNA mutations occur, single letter changes in DNA. And it's those kinds of changes that have to create de novo promoters if you want to be faithful to the biological process. And to meet that possible objection, we actually conducted now the second part of this experiment. And for the second part of this experiment, we started actually with random and genomic library sequences from our first part that are not already promoters. Okay? So this goes back to your question. There were actually a substantial part, about 85%, that were not already promoters. Okay? And tried to see whether we could create promoters from them by sprinkling them with random single letter changes on the DNA. So this is why the first part of this experiment was necessary to get to the second part. Okay, does that make more sense now? Okay. So the way we identify these are that cells that harbor them do not fluoresce in either red or green. And we call these sequences parent sequences. And we worked specifically with 16 random parents and 225 genomic parents. And from each of these parents, we applied a process that's called mutagenesis, where we basically use an enzyme called an error-prone polymerase. Doesn't really matter exactly what that does, but essentially it sprinkles a small amount of random changes, 1, 2, 3, or 4, into each of these 150 base pair sequence. And we performed this actually independently with each of our parents. So we basically created 60 plus 225 libraries of data sequences. And we call these mutagenesis libraries. So this is where this experiment becomes a whole lot of work, because we started with a single, the first part of the experiment was basically about a single library. Now we're talking about 285 different libraries. So schematically, what we did is shown here again. We started with non-promoter sequences, identified 225 genomic parents, 60 random parents within them, and then created multiple libraries from each, such that overall we created about $500,000. This is all shown as one pool of dollars, but this was done separately per library for the genomic parents, and $127,000 for the random parents. Cloned these again placed this again in our protoplasm, transformed that into E. coli cells, and then again used sort-seq to identify gene expression. Now again, the parent sequences we know don't actually drive expression at all, but we might find a situation like this here, where we have a daughter sequence that has a change from some nucleotide, some letter, to a C here, and that turns out to not do much to GFP expression, but it really turns on RFP expression. So this is, we would say, has created a promoter in the right to left direction RFP. And similarly, for some of the other sequences, here is one that has created 3.9 units of GFP expression. Here's one that didn't actually do anything, and so forth. And well, the most elementary question you can ask here is, what is the likelihood that a DNA mutation, that a mutated sequence like this creates a promoter? And we quantify, it's very easy to quantify once you have the data, through this quantity called P nu, is the probability that a mutation creates an active promoter. And it's simply the number of daughters that express GFP or RFP divided by the total number of daughters. And we calculate that separately for each parent of each library. The resulting data is shown here, summarized in a box plot, where each of these circles would correspond to data from one library. Overall, we find that 9% of the daughters of random parents are promoters, and there's a huge dispersion around that average. And that's another remarkable observation, I think, because it tells us it's actually fairly easy to produce by a very small number. In fact, single mutation show the same statistics, single letter changes, about a sizable fraction of new promoters. Yes, you had a question?
Yeah, did you allow for only one mutation in every?
No, you can't control that. So there's a distribution of mutations, but it's small. And in fact, the statistics are very similar whether you have 1, 2, or 3 mutations. And we see very few with more than 3 mutations. Now again, repeat that for genomic DNA. And here we find something that's the opposite of DNA. That is, genomic DNA is a bit less likely, in fact, 3 times less likely to, the new promoters are 3 times less likely to emerge from genomic data from random DNA. So these are the comparable values for P nu. And before I attempt to explain why that is the case again, let me just show you the kind of data that you get out of this experiment. So what you see here is very schematically a part of the GFP reported gene, part of the RFP reported gene, and here's only part of the 150 base pair insert. And it turns out that we have 1,919 daughter sequences in our library created from random parent number 10. So one of our random parents, where this part of the sequence is not changed. And although other parts of the sequence may have changed. And what we see is that this is essentially a violin plot of RFP expression, 1 being nothing, 4 being maximal, and that plot means there's basically no expression in these sequences. But it turns out we also have 30 library sequences where this particular nucleotide has mutated to a T, and that suddenly switches that promoter on rather dramatically. So we get close to maximal expression. So since we have a lot of data like that, we can analyze what really accounts for the turning on of expression in some of these sequences. And to make this a bit more quantitative, we used something that will be familiar with many of you. This is my only math slide. I'm not proud to say in this audience that basically we computed what's called the mutual information between nuclear identity and expression of fluorescence. The mathematical explanation definition of mutual information is shown here, where Pi of B is the probability that at position I of the data sequence you have a particular letter or base, A, C, G, or T, and P of F is the probability that a data sequence fluoresces at intensity F. And Pi of B of F is the joint probability. And what this essentially allows us is to quantify the likelihood that a change into a particular nucleotide in one of the data sequences turns on expression or turns a sequence into a promoter. And we can use it by actually averaging over all the positions I in such a sequence and averaging over nucleotides at each position to produce a mutual information profile that looks something like this. So this will be the length of one of our parent sequences. This happens to be random parent number 18. And here you see mutual information on the vertical axis. And you see this pronounced peak here that indicates that it's very likely that a nucleotide change in this region here causes expression. Okay? This dashed line indicates a significant threshold of what we consider a significant peak in this profile, which we also call a hotspot. That threshold is to some extent arbitrary, but we actually varied it and showed that most of our observations hold regardless of this threshold. So basically, we can use mutual information to identify which regions in such sequences are especially prone to become promoters. And here are some of the selection of some of the profiles, random and genomic sequences that we derived from this approach. Okay? It turns out this is already quite useful to help explain differences in p-nu among sequences, because sequences with more hotspots, that's shown here, a hotspot count per sequence, are more likely to turn into de novo promoters. And that holds for both for random and genomic sequences. Now, when you analyze our sequences with respect to these hotspots, we found that random parents contain more than 3 times more hotspots than genomic parents. And that's encapsulated in this highly skewed dialing plot of the distribution of the number of hotspots per parents. And that actually turns out to explain most of the difference between random and genomic parents. But it turns out we can do a bit more with that. And in particular, what we studied is what we call protoboxes. So a protobox is a DNA sequence that is 1 letter change away from a -10 or -35 box. Such that a single 1 letter change like this one would turn it into a -10 box. And here we can use, again, computational predictions integrated with this data. And it turns out random parents contain many more of these protoboxes than genomic parents. And this holds for both the -35 and the -10 boxes. And this explains why they harbor more hotspots. So it seems that genomic DNA, through whatever evolutionary forces, has evolved away from containing protoboxes, as if protoboxes were being suppressed in some way evolutionary, so that it's not easy to turn genomic DNA through single or small number of nucleotide changes into a promoter. Now, yes? Well, it's a good question, and we can talk about that afterwards. I think it would be a good discussion question.
Will it be a matter of kind of protection?
So that's the speculation that is on top of our minds too. The problem is to prove that. Okay, I think that would be a good question for a discussion. So I've told you so far about these boxes that constitute this. This is a messy slide. I will not get into it, but it's summarized here. I've also talked about that certain transcription factor binding sites or transcription factors may be needed to activate a gene expression, but it turns out they account for a relatively small fraction of our hotspots. That's because we repeated the analysis I just told you about with transcription factor binding sites. And for example, for random DNA, we find that most random parents become promoters by gaining either minus 35 bucks or minus 10 bucks. So this is these numbers here. But only 5.7% gain become promoters by adding and activating transcription factor binding site. You may notice that these numbers don't add up to 100%, and that's a reflection of our ignorance. That is, we don't know everything that's going on in these kinds of DNA sequences, which goes to show that computational tools still, after so many years of research on the E. coli genome and bacterial gene expression in general, is still very limited. Okay? So for the last 3-4 minutes, I want to talk about the opposite of activation, because everything we've talked about so far, the creation of minus 35 or minus 10 boxes or these transcription factor binding sites, was about activating sites, that is, sites whose creation causes transcription. But there's also the opposite, namely sites whose destruction causes transcription. We know, for example, in the E. coli genome that about half of the genes are activated and another half are repressed. So repression is at least equally important to activation. We call these sites repressing sites. And those would be sites where, that normally prevent transcription, but if we destroy them, then transcription may be possible. And we can analyze these kinds of sites as well. So we started predicted binding sites again for 93 different transcription factors and asked which of their sites are repressing. That is, when we destroy them, in our experimental data, fluorescence appears. Okay? And there we find that genomic parents harbor many fewer repressing sites. 0.12 repressing sites per genomic parent as opposed to 2.2 repressing sites per random parent. We can also stratify these in case anybody here is into prokaryotic gene regulation by potentially repressing transcription factors. So this is a list of transcription factor genes. On the vertical axis, you see the number of repressing sites per fragment for each of these factors. And in some of them, the difference is just staggering. We have many more repressing sites many more repressing sites in random DNA than in genomic DNA. We also see experimentally that destruction of a repressing site causes a much greater expression increase in a genomic parent than in a random parent. Now, to put this into context, it's important or useful to be aware that in many different organisms, expressing a gene where it's not supposed to be expressed is actually much more damaging or can be more damaging than not expressing a gene where it's supposed to be expressed. So this is called ectopic expression. And for example, developmental biologists in eukaryotes use it, people who study eukaryotic development use it to find out what a gene is really doing by expressing it everywhere. It turns out all kinds of damage happens in that case, but this damage can actually help us find out what the gene is doing. So what we think is what's probably going on here is that a very strong expression increase. So there's a fluorescence increase when repressing site is lost. Very strong such repression expression increase as a result of a destruction of a repressing site may have detrimental effects on E. coli. And this effect would be much weaker in the mutation or destruction of repressing sites in a random parent. Yes.
The destruction of this repression site. So, how many more mutations does it take? Is a single mutation?
Well, yeah, a single mutation is enough. Okay. So, what that gives rise to is the speculation that perhaps, as natural selection eliminated all those repressing sites from a genome. And we have some tentative evidence for that, in the form of the distribution of the number of repressing sites in our fragments. So again, what you see here is a plot which we see here the number of repressing sites stratified by information content again. And this is basically what we see for random DNA. And this is what we would expect by chance alone. So this is random DNA by definition. But if you look at genomic DNA, that distribution is much flatter. So it means that even there's fewer repressing sites in the genome if you look at the overall distribution, and that holds especially for low information content sites. So what this suggests is that there's some evolutionary force. We cannot prove that it is natural selection from the data that we have, but some evolutionary force that has changed the distribution to reduce the fraction of repressing sites. Okay? Beyond that, we don't know what causes this difference. So I'm at the end here, and very good. We are exactly at 1 hour here. So I'll just summarize for you. We found that an astonishing proportion of random DNA fragments harbor active promoters. And that genomic DNA is more likely to harbor promoters than random DNA, even inside genes. Conversely, random DNA is 3 times more likely to give rise to promoters than Novo. And the fundamental reason is that genomic DNA is impoverished in promoter sites. Genomic DNA also contains fewer repressing sites than random DNA, that is, fewer sites than expected by chance. And their destruction, we find, leads to much greater activation in genomic DNA. So overall, it looks like to us that something has impoverished genomic DNA in promoter sites and has impoverished also in repressing sites. All those kinds of sites that, when changed by a little bit, could cause expression of DNA that is not normally expressed. Now, the fundamental question that we have at this point is, can we prove which evolutionary forces cause these differences between genomic and random DNA? And some of you have speculated that, maybe it's selection, and selection wants to favors the suppression of the existence of such sites. And if this is indeed the case, would lead to a very interesting conclusion. And what this would tell us probably is that, in contrast to what early biologists like François Jacob thought, namely that it's very, very hard or perhaps impossible to create genes or elements of a gene de novo, it turns out it's so easy that evolution may actually have to suppress it. It's extremely likely, okay? But we don't know that yet for sure. It's only that we have some intriguing hints towards that. I thank you for your attention, and if you have any more questions, here I am.
So that last part kind of conflicts with the observation number 2, right? So it's suppressed. So these proto boxes are suppressed. But
Yes.
the formed promoters are actually enriched.
Exactly. So
Are they only in introns there?
there is no introns in E. coli. Okay. So, I think I made a disclaimer sometime during the talk. We do not understand all the patterns that we see there.
Yeah.
Okay. And I think there is a lot of interesting work to be done by theorists and computational data scientists, because all that data is going to be public.
Oh.
Yes.
I have a question when you say the beginning, like it's random and have never been known that it's a promoter. Is that like they never known for the E. coli or never been known in a closest species, genus or something?
So, when I talked about random DNA at the beginning, what I said is that it's DNA that has never been inside an organism, right? But maybe there's some organism out there that happens to have a piece of DNA that is identical to the one that's coming out of our DNA synthesizer, right? But the likelihood of that is not very large. You could probably, not sure whether you work with biological data, you could probably blast our entire library against genomic DNA that's been sequenced. I suspect you wouldn't find much similarity out there. Does that answer your question?
Yes.
Okay. The 2 ladies in the back, who wants to go first?
You mentioned as well that some genes don't have the -35 and then 10 blocks. How would that go then? Or have to look them to that?
We have no idea. So, I have presented a sort of a simplified view of a bacterial promoter. There's all kinds of other things that are going on, even in something as simple as bacteria, right? If somebody, for example, like yourself, were able to identify a sequence signature that characterizes uniquely and sufficiently, and so that was necessary and sufficient for, to define a promoter, you'd probably see that in our data. So the data could be analyzed for that. We just didn't find the right way to do that. Yes.
Yeah, it's also been a related question. So I wonder how good is our knowledge now? Let's say you took your random library and you would, with the knowledge you have beforehand, predict which ones are promoters. How much % would you give?
I wouldn't even go that route. It would be depressing.
Depressing in what way?
Depressing in a way, so something that's well known both from prokaryotes and eukaryotes, that if you predict transcription factor binding sites, this is where it's been done more actually, you'd find hundreds of transcription factor binding sites for most transcription factors, but maybe a dozen or so or 20 or so regulate the gene. So there's tons of false positive results, and this is that you get computationally, and this doesn't change any of that. I'm not sure, Tuan, perhaps?
Yes, I have a question regarding the last comment of the question about this evolutionary process. Can I speculate some role of energetic cost here? For instance, on this transcription process involves some energy cost, and then maybe optimization for metabolic efficiency in one of the evolutionary drivers. Is this something that I can speculate?
Yeah, so we know that in organisms like E. coli, there's sort of population genetic theory that it has very large population sizes. So natural selection can detect, if you will, very, very small costs of any process that goes on. And in an organism like E. coli, this kind of energetic cost of expression is a candidate for what might suppress or what might cause damage if you turn on the transcription of a particular part of a genome that shouldn't be turned on. My suspicion, though, is that actually other cost components will be more important. For example, if you transcribe part of a gene from inside the gene, that may interfere with the correct way of transcribing the gene, namely starting from outside the gene. Okay, in fact, we know examples of genes that are regulated in that way, where the gene is normally described in one direction, but there's a small transcript that's made in the opposite direction from inside the gene that causes a double-stranded RNA to form that can then no longer be translated properly. So that's a known mechanism of gene regulation. So I would imagine that those kinds of costs, where there's active damage caused by ectopic gene expression, would be more important than energetic costs. Yes.
I was wondering, so all of what your results are that you give rise to new promoters, but do you also see the opposite, that you stabilize existing promoters?
Yeah, so it's a very good question, but we have not looked at that explicitly, and part of the reason is, well, let me ask you, how would you define the stabilization of a promoter? When has a promoter become stabilized?
I know, I'm not I guess that the intensity of expression, I would say.
Yeah, so what we do know, and we know this from, we have a paper on this, but it's just basically on computational predictions, that promoters that tend to be stable, and I'll define stable for you for a moment. So it's a promoter that cannot simply be disrupted by a single mutation, right? We call those promoters not stable, we call them robust, but that's just a matter of semantics. Those tend to be very strong promoters. So the strongest promoters are also the most robust promoters. Yes?
Thanks, Bob. Thanks, Brad. So maybe I'm not worried about the question, but there was like in semantics as well about algorithmic information theory, whether you consider there this compressibility of genomic DNA versus compressibility of random DNA.
So we have not looked at this here at all, algorithmic compressibility. I would suspect that genomic DNA is more compressible than random DNA. But I couldn't really defend that prejudice if you asked me to.
Maybe not big.
Sorry, let's have it first.
Yeah, it will seem to be not really about more complicated theoretical science thing, but just if you compare your random DNA, the genomic DNA, there are statistical tests you can probably do it, like how often do you see certain letters together or
Yeah. People have done this up and down, and
Yeah.
the interesting thing is that I'm sorry, I didn't ask.
Well, I think it was like, that wasn't sort of quick, like, can you use some sort of, there is a lot of difference between genomic and random DNA, but it doesn't help you in.
Unfortunately, it does not.
No.
And also the statistical structure of DNA, the last time I looked, was essentially an unsolved problem. That is, there is no, DNA has short-term correlations of between nucleotides of, quite close together and very long-term correlations. And some of the best statistical experts in the world, for example, Samuel Carlin at Stanford, is known for his work on stochastic processes. Have worked on this problem for years and have not been able to come up with a satisfactory statistical model of genomic DNA.
But could you, for example, generate random DNA fragments that have this short range, same short range?
Yes. You could do that. And when you actually design such an experiment, the first thing you agonize about is how random should you make your random DNA? And it turns out, in which way would you want to make it similar to genomic DNA, right? And the problem is that, well, there is only one way in which you can make random DNA IID, identically independently distributed across nucleotide positions, but there is a bazillion ways in which you could make it resemble correlations, right? That, for example, you could manipulate the AT content. You could manipulate the dinucleotide distribution or trinucleotide distribution, and you could go up to any order, right? So the number of possibility is just combinatorially prohibitive. So we stuck with the simplest thing.
But so if I understood you correctly, the promoters are generally TA rich,
Yes.
right? And you said that your random DNA is 25% ATG. So then, do you already then have a bias?
Yeah, so this is something we worried about a lot, and the fortunate thing is, and this is why I mentioned this without saying it really explicitly, is that we actually have a distribution of AT content in the random DNA because we have so many fragments, right? And that distribution is sufficient to actually also cover the AT content that we see in E. coli. So even with the AT content window that exists in E. coli, that's characteristic of E. coli, we have enough sequences in the random library to do statistics. Okay? And if we had not 10,000 but 10 million sequences, the statistics would be even better. But I think it's sufficient for what we wanted to show. Okay?
But the AT content of E. coli is lower than 25%.
The AT content of E. coli is somewhere around 54%. So AT, if it's random, it would be 50%. Right? So there is genomes that are GC biased and AT biased, okay? And we still don't fully understand why that is the case. It's not an interesting fact, right? But it's not very extreme. So it's not extremely different from our random DNA. You had another question, or you
Yeah, I have a speculative question. So, as far as I know, nobody really knows how promoters promote transcription. Does this give you any? Does this new sort of non-discrete view on promoters bias towards any of the theories, like this liquid phase separation story? Do you have a tendency now to say, this actually fits this one explanation of promoters well?
It's a good question. I have not thought about it. Okay. I would also say that what you say holds more so for eukaryotes than for prokaryotes. I think there's some very good biophysical models of promoters in E. coli. So it's something that's very, very interesting, for example, with how the RNA polymerase finds a promoter.
Exactly.
Namely, it binds at a random place and then it diffuses linearly, random Brownian motion, until it hits the nearest promoter, right? And then it stays there and it starts transcription, right? So that process, I think, is fairly well understood. I'm not sure that, you know, the level of resolution in our data is, it tells us a whole lot about the biophysics of, or biochemistry of protein promoter regulation. Yes?
There is one question from Peter.
Oh, sorry, yes.
I mean, I'm also interested in nucleotide compactification, so DNA compactification for
Yes.
associated proteins. Can you say something about the role of compactification on results? Do they play a role?
They could well play a role. It may be one of those many factors.
But sorry, I don't understand. the question.
Compactification.
Oh.
So it's the fact that, you know, whether in eukaryotes, the process is, you know, well understood, but in prokaryotes too, prokaryotes, it exists as well. The DNA is, you know, not just, you know, sort of this free-floating molecule that is, you know, it's very compact, and it's often compactified even more by proteins, right? And that could well have a lot of ramifications for example, our inability to predict promoter sequences that are functioning just from computational data, because there may be subtle sequence signatures in a genomic region that influence compactification and may prevent expression of a particular sequence, and what the sequence of a -35 or -10 box may tell us nothing about that.
I have
Yes?
many questions. So in this figure of this mutual information profile,
Yeah.
I would have expected there to be peaks at -10 and -35. And I didn't see those.
So that's actually a very good point. Now, we don't know actually where +1 is in these fragments, in this DNA. So, you know, a transcript could start in a different place from where GFP, for example, is normally transcribed, right? It could start further upstream or even a bit further downstream, right?
Okay, right.
We have not mapped the transcription start for any of these. That would be an entire another 4-year project.
Any other question? If not, then can Andreas.